Welcome to this lesson on droplet-based directed evolution. Today we're going to explore one of the most powerful technologies in modern biotechnology, how we can harness millions of tiny microreactors to evolve enzymes and cells with extraordinary speed and precision. Let's start with the fundamentals. What exactly is directed evolution? Directed evolution is a method that mimics natural selection in a laboratory. Instead of waiting millions of years for nature to optimize proteins, we accelerate the process to days or weeks. The traditional approach involves 3 steps. First, create genetic variants through mutation. Second, screen each variant for the desired activity. Third, select the best performers and repeat the cycle. But here's the fundamental problem. If you want to screen millions or billions of enzyme variants, traditional microtiter plate methods are impossibly slow and expensive. A single 96-well plate takes hours and costs dollars per sample. This is where droplet microfluidics comes in. It's a revolutionary technology that changes everything about how we conduct directed evolution experiments. Imagine a microfluidic chip, a small device with microscopic channels etched into glass or plastic. Aqueous droplets containing cells or enzymes flow through these channels, surrounded by an immiscible oil phase. Each droplet is a tiny, isolated reactor. Each droplet is incredibly small, typically 10 to 100 picoliters in volume. That's 1/10 billionth of a liter. These droplets serve as independent microreactors. Here's where it gets impressive. These droplets can be generated at rates of 1,000 to 10,000 per second. That means millions of experiments per hour. Each droplet is completely isolated from its neighbors by the oil phase. This means there's no cross-contamination between reactions. Each variant gets its own private reaction chamber. Let's do a back-of-the-envelope calculation to understand just how much faster this is compared to traditional methods. With traditional methods using 96-well plates, you can process 96 samples in about 4 hours of work. That's 96 divided by 4 times 3,600 seconds, which equals 0.0067 samples per second. Now compare that to a droplet system generating 5,000 droplets per second. The speed increases 5,000 divided by 0.0067, which is approximately 750,000-fold faster. That's nearly 1,000,000 times faster. To screen 1,000,000 variants in droplets takes only 200 seconds. That's about 3 minutes. With traditional methods, this would take years. Now let's understand the physics of how these droplets are actually generated. The most common method is called T-junction droplet generation. An aqueous stream meets 2 oil streams at a T-junction. Surface tension and shear forces break the aqueous stream into uniform droplets. The droplet size is precisely controlled by the flow rates of the aqueous and oil phases, as well as the geometry of the microfluidic channels. This gives us exquisite control over reaction volumes. Special surfactants are added to the oil phase to stabilize the droplets. These surfactants prevent droplets from merging together, which would destroy the isolation we need. Here's a key question. How do we ensure that each droplet contains 1 cell or 1 genetic variant? We use Poisson statistics for single-cell encapsulation. If we dilute our cells to the right concentration, we can control the probability that each droplet contains exactly 1 cell. The Poisson distribution tells us the probability. P of k equals lambda to the k times e to the negative lambda, divided by k factorial, where lambda is the average number of cells per droplet. For example, if lambda equals 0.1, meaning an average of 0.1 cells per droplet, then p of 1 equals 0.1 times e to the negative 0.1, which is approximately 0.09 or 9%. About 90% of droplets are empty, and only 0.5% have 2 or more cells. This ensures that most occupied droplets contain exactly 1 cell. For enzyme evolution, we don't need whole cells. We use a technique called in vitro compartmentalization, where we encapsulate DNA, transcription translation machinery, and substrate all in 1 droplet. Now we arrive at the most crucial part. How do we sort millions of droplets based on the activity of the enzyme or cell inside? The sorting system works like this. Droplets flow single file past a laser beam. The laser excites fluorescent molecules in the droplet. A detector measures the fluorescence intensity. Based on this signal, an electric field deflects high fluorescence droplets into a collection channel. We use fluorescent substrates that only become fluorescent when acted upon by the enzyme. Higher enzyme activity means more substrate conversion and brighter fluorescence. This is similar to fluorescence-activated cell sorting, or FACS, but instead of cells, we're sorting droplets. We can sort at rates of 1,000 to 5,000 droplets per second. The detection happens incredibly fast. Each droplet is interrogated in just 10 to 100 microseconds. This speed is what enables the massive throughput. Let's look at a concrete example. Researchers have used droplet-based directed evolution to evolve horseradish peroxidase, an enzyme used in diagnostics and biotechnology. The goal was to increase the enzyme's activity and stability. They wanted a peroxidase that works better under harsh conditions. They created a library of 10 million variants through random mutagenesis. Each variant was encapsulated into droplets with a fluorogenic substrate. After incubation, they sorted the droplets and collected the top 0.1%. From 10 million variants, collecting the top 0.1% means 10,000 variants are kept. After 5 rounds of evolution, they obtained enzymes with 200-fold improved activity compared to the starting enzyme. This would have been impossible with traditional screening methods. The entire directed evolution campaign took only 2 weeks. Using traditional methods with microtiter plates, this would have required at least 2 years of continuous work. A critical question arises. After we sort droplets based on fluorescence, how do we recover the genetic information from the winning variants? For cell-based systems, this is straightforward. The cell carries its own DNA. After sorting, we simply grow the cells and sequence their genomes. For cell-free enzyme evolution, we ensure each droplet contains the DNA encoding the enzyme along with the enzyme its. After sorting, we recover and amplify the DNA using PCR. Sometimes we use water in oil and water double emulsions. This allows us to break the outer oil shell and pool the inner aqueous droplets for DNA recovery. Modern systems can do even more sophisticated sorting. Let me explain multi-parameter sorting. We can use multiple fluorescent colors simultaneously. Green fluorescence might report enzyme activity. Red fluorescence reports stability under stress. Blue fluorescence reports substrate specificity. We can then sort based on combinations. For example, select only droplets with high green fluorescence and high red fluorescence. This lets us optimize multiple properties at once. This simultaneous multi-parameter optimization is extremely powerful. In nature, organisms evolve one trait at a time over eons. Here we optimize multiple traits in parallel. Let's address an important question. What limits the diversity we can explore? A codon is 3 DNA bases. With 4 possible bases, that's 4 cubed equals 64 possible codons. For a 100 amino acid protein, there are 20 to the power of 100 possible sequences. That's about 10 to the 130, an incomprehensibly large number. This number, 10 to the 130, is vastly larger than the number of atoms in the observable universe, which is only about 10 to the 80. In practice, we can screen libraries of 1 million to 1 billion variants using droplet microfluidics. This is still a tiny fraction of sequence space. The solution is iterative evolution with smart library design. We don't randomly search all of sequence space. We focus mutations around known functional sequences, gradually walking uphill in fitness landscape. Droplet-based evolution isn't just for enzymes. We can evolve entire cells for desired functions. For example, we can evolve bacteria to produce biofuels more efficiently. Each droplet becomes a tiny fermentation vessel. We sort based on fluorescence from a sensor that detects the desired product. Or we evolve yeast for tolerance to high alcohol or temperature. Cells are exposed to stress conditions inside droplets. Only surviving cells produce fluorescence. After stress exposure, we use a viability dye that fluoresces green in living cells, but red in dead cells. We sort to collect only the green droplets containing survivors. We can even monitor cell growth inside droplets over time. Droplets with faster growing cells become more turbid or show more fluorescence from a constitutive marker. Let me summarize the key advantages of droplet-based directed evolution over traditional plate-based methods. First, speed. As we calculated, it's roughly 1 million-fold faster than traditional methods. This enables screening library sizes that were previously impossible. Second, cost. Picoliter-scale reactions use 1,000 to 1,000,000 times less reagent than microtiter plate assays. This makes expensive substrates or enzymes affordable. Third, isolation. Each variant is tested in its own isolated compartment. There's no risk of cross-contamination or cheating, where one cell benefits from the product of another. Fourth, single-cell resolution. We measure the phenotype of individual cells, not population averages. This reveals rare high performers that would be missed in bulk assays. Fifth, quantitative measurements. Modern detectors can measure fluorescence with great precision, allowing fine discrimination between variants with subtle activity differences. Of course, this technology isn't without challenges. Let's discuss some key technical hurdles and how they're addressed. First challenge, droplet stability. Some reactions require hours or days of incubation. Droplets must remain stable without merging or breaking. The solution involves carefully optimized surfactant formulations and biocompatible oil phases. Researchers have developed fluorinated oils and specialized surfactants that keep droplets stable for over a week. Second challenge, getting substrates into droplets after formation. Sometimes we want to add a substrate after cells have grown in droplets. The solution is pico-injection. Droplets can be merged with smaller substrate-containing droplets using electric fields. This allows multi-step reactions in the same droplet. Third challenge, false positives. Sometimes cells or enzymes produce fluorescence through non-specific mechanisms unrelated to the desired activity. The solution involves counter-screening strategies. We use multiple fluorescent substrates or perform secondary validation assays on sorted variants before the next evolution round. Let's calculate how much enrichment we get in a single sorting round. This helps us understand how many rounds we need. Suppose we have one good variant hidden among 1,000,000 mediocre variants. The starting frequency is 1 in 10 to the 6. We sort and collect the top 0.1%. That's 0.001 times 10 to the 6, which equals 1,000 droplets collected. If our good variant is in that top 0.1%, we've enriched it by a factor of 1,000. It went from 1 in 1,000,000 to 1 in 1,000. After 3 rounds of 1,000-fold enrichment each round, we achieve 1,000 cubed equals 1,000,000,000-fold total enrichment. Our rare variant is now the dominant sequence. Let me describe a cutting-edge development. Not all enzymatic reactions produce fluorescent products. What if we want to detect absorbance changes? The problem is that absorbance measurements typically require longer optical path lengths than the diameter of a picoliter droplet. A 50-micrometer droplet gives very weak absorbance signals. The solution involves ultra-sensitive detection using optical cavities or integrated waveguides that increase the effective path length through the droplet by multiple passes. This enables screening of oxidases, reductases, and other enzymes without requiring fluorogenic substrate analogs. We can use the natural substrates of the enzyme. Let's talk about real-world impact. Droplet microfluidics is revolutionizing pharmaceutical drug discovery. Pharmaceutical companies use droplet platforms to screen billions of drug protein binding events. Each droplet contains one compound from a chemical library tested against a target protein. For antibody discovery, they can screen 100 million antibody variants per day. This accelerates the identification of therapeutic antibodies against cancer or infectious diseases. Traditional high-throughput screening processes may be 10,000 compounds per month in a large facility. A droplet platform can screen 100 million compounds per day. That's 10,000 times faster than traditional methods. There are also environmental applications. Researchers are using droplet evolution to develop better bioremediation organisms. They evolve bacteria that can degrade plastics, petroleum compounds, or environmental toxins. Each droplet contains bacteria and the pollutant substrate. Fluorescent reporter systems indicate when the pollutant has been successfully degraded. High-activity variants are sorted and enriched over multiple rounds. A success story is the evolution of PEtase enzymes that break down polyethylene terephthalate plastic. Researchers achieved 100-fold improved activity using droplet-based directed evolution. Looking to the future, an exciting direction is integrating machine learning with droplet-based evolution. Machine learning algorithms analyze the sequence-function relationships from previous rounds. They predict which regions of sequence space are most promising to explore next. This enables smart library design. Instead of random mutagenesis, We focus mutations in regions predicted to yield high fitness variants. This dramatically reduces the search space. Eventually, we'll have fully automated closed-loop systems. The machine designs a library, robots build it, droplets test it, and the machine learns from results and designs the next round, all without human intervention. How does droplet microfluidics compare to other high-throughput directed evolution methods? Phage display is powerful for antibodies, but limited to proteins that can be displayed on phage surfaces. It can't evolve intracellular enzymes or whole cell phenotypes. Yeast display, combined with flow cytometry, can screen about 10 million variants, which is good but still less than droplet systems that handle billions. Ribosome display is entirely cell-free, but lacks a physical compartment. This makes some assays difficult, especially those requiring multiple reaction steps. Droplets are the most versatile. They achieve the highest throughput and can accommodate almost any type of assay: enzymatic, binding, whole cell metabolism, or growth selection. Let's discuss practical economics. Is this technology accessible or only for large institutions? A basic droplet generation and sorting system costs about $50,000 to $100,000. This includes pumps, microscope, cameras, and sorting electronics. Commercial automated platforms cost $200,000 to $500,000. These are turnkey systems with software and support. But here's the key advantage. Reagent cost per million assays is only $50 to $100. With traditional methods, 1 million assays would cost millions of dollars in reagents alone. For an active research lab doing directed evolution, the return on investment is less than 1 year. The savings in reagents and labor quickly offset the equipment cost. We should also consider ethical and safety aspects of this powerful technology. From a biosafety perspective, droplet-based evolution is actually safer than some alternatives. The entire process happens in contained droplets. Evolved organisms aren't released until they're characterized. However, there's a dual-use concern. The same technology that evolves beneficial enzymes could theoretically be used to evolve harmful organisms or toxins. This requires responsible oversight. The scientific community is developing guidelines for responsible innovation. Training programs teach not just the technical skills, but also the ethical framework for using these tools. Let me summarize the key concepts we've covered in this comprehensive lesson. Droplet microfluidics creates millions of isolated picoliter-scale reactors, each serving as an independent experiment. Fluorescence-activated sorting enables screening of billions of variants at rates up to 10,000 per second. This represents a 100,000 to 1 million-fold speed increase over traditional microtiter plate methods. Applications span enzyme evolution, whole cell engineering, antibody discovery, drug screening, and environmental bioremediation. The technology enables exploration of sequence spaces that were previously completely inaccessible, opening new frontiers in protein engineering. Integration with machine learning and laboratory automation promises even more revolutionary advances in the coming years. Here's a final thought to leave you with. Droplet microfluidics is democratizing directed evolution. What once required massive industrial-scale facilities and millions of dollars now fits on a desktop and costs thousands of dollars. This empowers small, academic labs, startups, and researchers in developing countries to make breakthrough discoveries that were previously only possible for large pharmaceutical companies. We're accelerating the pace of biological innovation worldwide. The next breakthrough enzyme, therapeutic antibody, or bioremediation organism might come from a small lab using droplet technology. The future of directed evolution is quite literally in these microscopic droplets. Each one is a tiny laboratory where nature's creativity meets human ingenuity to solve some of our most pressing challenges.